Ladies and gentlemen, um, you would like me to have the microphone? Yes. God damn! Is my voice not loud enough? Really? Damn. Hi everyone, now I have to lower my voice. Welcome and thank you for sticking around for the final session of day 2. What are we talking about? That's a very good point. Point of care and real-time medical care and utilization of biomarkers. So we just had the session on biomarkers, and now how can we use... Is this the other? How can we use them? How can we use those biomarkers, and what could we create? What point of care devices might we create? So we have an interesting lineup. We have a PhD student giving a talk on microimmune, that's the project title. We have some chemiluminescent biosensors, I hope. We have industry is here. Oh yes, that is very exciting. They are the ones that are helping create these devices and to get them flying. So boy, that's exciting. And finally, I hope we're having also a talk about blood coagulation in space flight related environments. Anyway, I'm going to stop talking and hand it over to our first speaker, who is Marina Tushin. Thank you very much. Ladies and gentlemen online, welcome back. Next up, I hope we have Mara, but I haven't. Ah, hello. Mara, are you talking about chemiluminescence biosensors? Fantastic. Yep.
Okay, thank you. So thank you for giving me the opportunity to speak about our recent and ongoing activities in the development of biosensors. I removed chemiluminescence because I added a small contribution at the end. First of all, I want to point out that here I put only my name because there's a full list of people contributing. So I will have a slide with the list of all the people contributing at the end because it wouldn't fit here. We had a very wonderful introduction about application of miniaturized analytical devices in space, and we are in a point-of-care testing session, so we all know that point-of-care testing can be very useful in space because we expect the human body to be to require monitoring and early intervention for any outcome that could happen during the mission. And so we need to develop and to obtain point-of-care testing devices that can be operated on space. And indeed, in clinical chemistry and in diagnostics, many diagnostic information comes from the analysis of biological samples and the quantitative and sensitive detection of biomarkers. So we need analytical tools to perform these assays directly on board the spacecraft. And so with this, in this context, the development of miniaturized analytical devices and especially of lab-on-chip devices can be very a good approach because they provide a high degree of integration and miniaturization possibilities and automation. But when we think about deploying analytical devices in space, we need to try to offer many positive features. So we shall try to develop reusable devices that can be refurbished and easily reconfigured to perform several consecutive analyses and with reduced consumption of resources. If possible, we should try to develop devices that can be very versatile in their application. So find different possible application in different contexts during the campaign to reduce, again, resources. And of course, they need to be operable in space, so gravitationally independent and resilient to space environment. And they need to be as much as possible automated to reduce crew time use and to reduce dependence on Earth. So we had the possibility to try to give our contribution in this field through two projects that are supported by the Italian Space Agency. And the first project was in situ bioanalysis, was performed in the recent years, and it was about providing a first example of an immunoanalytical device that would be employed on board the ISS for analyzing biomarkers in saliva, and that was used on board the ISS in 2017 by Paolo Nespoli. And now we are working at an improved version of the analytical device, going to the lab-on-chip technology and trying to provide additional functionalities. We decided to work on saliva because it is easily non-invasively collected biosample in which a number of biomarkers are established and can be used for diagnostic applications. And in particular, we focused on salivary cortisol, a stress biomarker in the first project. And now we expanded to cortisol and the hydroepiandrosterone, which can also provide information about immune system dysregulation. So our approach is, again, on immunoassays, but we exploit chemiluminescence detection. Why immunoassay? Well, we have seen a wonderful presentation, and immunoassays are a reference analytical technique for many diagnostic biomarkers. It provides very sensitive and specific detection. And we exploit the chemiluminescence type of detection for immunoassay. In this case, we use an enzyme as the label, and we exploit chemiluminescent reaction, which is a luminescence phenomenon in which photons are produced from by a chemical reaction. So photons will come out from the dark, and this will make the signal-to-noise ratio very favorable to reach low limits of detection and to obtain high detectability in very small volumes, which is very useful when we want to miniaturize the assay. So in first project, the in situ bioanalysis, we actually didn't use lab-on-chip technology. We started from the lateral flow immunoassay, which we all know very well since the pandemic, but because it's very convenient, easily used and rapid, and it relies on capillarity, which is good for microgravity condition. But in the conventional format, lateral flow immunoassay is visually observed, so you can only obtain yes or no response, while we coupled this format with chemiluminescence detection to obtain quantitative information. And in order to allow performance of the assay in a safe and easy way, we developed an analytical cartridge that is disposable and that contains all the reagents required for the analysis that would be easily operated by the crew in a manual way. And we also provided a easy to use and safe handling system for collecting saliva and injecting it into the cartridge and perform the analysis, and the chemiluminescence signal would be detected employing a CCD camera-based detector that would obtain a digital image of the photons emitted that would be analyzed to provide analytical information. And this is the assay principle. So we, in the cartridge, we mixed the saliva that was just injected with an immunoreagent, and these would flow along the nitrocellulose strip where we have immobilized antibodies that would capture biomolecules. And in the subsequent phase, we flow the chemiluminescence cocktail that would elicit a photon emission in correspondence of the lights, which intensity is proportional to the analyte concentration, so we can obtain quantitative information from the digital image. And okay, this is good. And this is a video, short video about Paolo Nespoli performing the experiment on board the ISS. So he would collect a saliva sample and then inject through a syringe the saliva sample into the cartridge. But this activity, I should mention, was requiring quite a lot of crew intervention. It was totally manually operated, so that's why we are now going to the second version to automate it. It was, nevertheless, it was quite straightforward and easy to perform, and we had successful experiments, so we were very happy with this first example. And then the CL reader was on board the ISS for quite a long time. We could also see that CCD is not, at least this type of CCD was not very resilient for long-term permanence on board the ISS. So now we switched to a different type of technology. First of all, well, this is the ongoing project. First of all, we want the system to be totally automated to reduce crew time use, and we, in collaboration with, oops, sorry, too much. Collaboration with engineers, we now integrate into the microfluidic chip thin film photosensors, which are based on amorphous silicon, and which is much more resilient to radiation, so they can work for a long time with no reduced damages. And so why am I sorry? I'm pushing the wrong button. So this is the picture of the system that contains all the fluidics, the microfluidic chip and the actuators, and it is connected to a disposable cartridge that contains all the reagents for the analysis, and that would also collect all the wastes and be safely trashed at the end of the procedure. And in order, since this microfluidic chip is integrating thin film photosensors, we wanted to make it reusable for multiple assays. So instead of immobilizing antibodies directly on the walls of the microfluidic chip, we are using magnetic microbeads functionalized that would be trapped in the position during the assay, and then released and washed away to have full chill reusability for many subsequent analysis, because we don't want to trash this technology, which is precious to us, a resource. And we can also, we are now working on 2 analytes, but it is scalable to a higher degree, a high number. So we can trap beads for different analytes in different positions along the channel, and each bead will have its own photosensor dedicated for its analysis. And at the end, we wash them away. And by changing the disposable cartridge, we can potentially analyze many different biomarkers in the sample. Okay, this is the add-on at the end. I thought it would also be useful for the discussion to bring our ongoing activity also in the development of wearable sensors. This is a completely different technology. We, in collaboration with many groups, so we are working at the development of organic electrochemical transistors, sensors that are integrated into a smart bandage. And we are applying this in collaboration also with Monica to the monitoring of wound healing biomarkers for obtaining diagnostic information. So we are now working at the development of a wearable patch that would be equipped with a series of electrochemical transistors, each one targeting a different biomarker. And in particular, again, we are working at functionalizing these electrodes with antibodies or with enzymes to provide biospecific detection of the biomarkers of interest. And there is a group of engineers who is also developing the front-end electronics for the signal elaboration and transmission. So again, as I mentioned, this is the list of all the people who are contributing to these projects, which I really want to thank. And so to conclude, I think that it's quite straightforward to agree on the fact that developing point-of-care testing devices is very useful for space, but also for its possible application on Earth. These technologies' advancements can be good also for applications of point-of-care testing on Earth. And thank you very much for your attention.
Do we have some questions? Have you got one?
Yeah, so thank you, first of all, for a wonderful presentation, excellent work, and looking forward to see your next version fly. So can you allude a little more about the exact test that you can do?
Yeah. Oh, okay. With the Aphrodite, the one that
The new one, the new...
Yeah. Okay. Now, at the moment, we are targeting cortisol and the hydroepiandrosterone in saliva, because we wanted to stay with the saliva sample, which is easier to handle also on the device. And so there are not so many well-established biomarkers in saliva. Potentially, we could go to other samples, but that would also require a sample pretreatment unit, which we don't have at the moment. So we hope for the future to be able to develop and to have opportunity to continue working on this development.
Yes, I think that's also what I had in mind, because I saw those two and I thought, okay, as a clinical physician, also in the emergency department, also in space medicine, when would I do these tests, right?
Yes.
We do tests because we want to make a differential diagnosis. So I'm not saying these are not important, and also for scientific reasons, there's definitely value in this. But my suggestion is really think about the next iteration to what tests you want to are the highest value, the most bang for your buck, as we say, to give us a good differential diagnosis. For example, is it an infection or is it not an infection? So the stuff that we deal with in real life clinical medicine as well, that gives us a better direction for medical care.
I know we have more questions, but we're going to move on for time. Thank you very much. Ladies and gentlemen, up next we have Mikhail Twisk, who's discussing a range of point-of-care projects. Go ahead.
All right. Okay, so I guess I am the representative from industry. First of all, I'm very humbled to be here. My background is in product development and technology development for I was going to say space applications, but actually for on-Earth applications. So in the last, let's say, 24 hours, I've been blown away about the research that you are all doing. And I'd like to give you a little bit of insight of where I come from and potentially inspire you as you've been inspiring me. But first, a quick introduction. I am from industry, so there's always a little bit of maybe a bit more commercial side to it as well. So Verhaert is a company that originated in 1969 and in the kind of 80s started developing satellites as a side hustle. That became a little bit bigger and eventually in 2004 we split that off to Kinetic, which has now been acquired by Redwire, and that's our history related to space. But in general, we're focusing on physical product development as well as strategic innovation. I have to say, within our department of high tech, we do still work with European space projects, for instance, for a project for the 3D bioprinting solution, as well as a project for monitoring or investigating shear flows on blood. But that's not me. I work in product innovation, and I am responsible for medical and life science projects. Quickly, we are an innovation company. We have a space solutions department, which currently develops with ESA on the two projects I just mentioned. But we also run incubation centers, and we help bring startups to either space applications or to translate space science towards Earth applications. So people that are in the room have worked on a space project that are interested in understanding, like, how can I valorize that and build a commercial company on Earth? That's also possible. a couple of examples, but I'm not going to go into that because that's not relevant for today's talk. And then I finally wanted to mention quickly that if you're from Belgium and you have an idea for a space mission or a space related application using technology that is available on Earth, then there's a current funding scheme, and I heard several people mention funding in the last couple of days, but there are effectively also funding schemes that go very bottom up. All right, so that was all about topics that I'm not very comfortable with. What I am much more comfortable with is bringing products to the market for life science applications, and for me that means medical devices, but also devices that can be used for drug manufacturing or drug discovery. And I think in the last day I've noticed that actually point of care is a big item. We want to be able to monitor our astronauts from the perspective of kind of a research element, where we want to understand what is actually happening to my astronaut in space, because there's so many factors that we can't simulate on Earth. We need to do that in space, and I hope I can shine light on some innovations that are currently being developed on Earth that could be used in space to monitor the astronauts, but also similarly, we want to keep them healthy like we keep our patients healthy or humans healthy on Earth, and how are we going to provide them with the care that they need. And I think we have a lot of challenges actually on Earth to do so, so I can imagine that it is much, much harder to do that in space. And so I'm gonna try to inspire you. I'm not allowed to talk about the kind of data that we gather on the development of these devices. These are confidential, but I like to give you a bit of an idea of what type of developments are going on, because I think they might be relevant for you. And this is the first example. Is actually the ability to develop flexible electronics, and don't ask me how they perform under radiation, but that also allows us to make flexible wearable patches. And one of the flexible wearable patches that we think is very interesting is a kind of large area ultrasonic patch. And that is interesting for us as human beings that are focused on Earth applications, to monitor patients that are in the ICU, and that we want to be able to monitor whether their heart continues to perform as good as they do, because the ECG actually isn't a very direct measure. It's more an auxiliary measure of the heart's performance, and we can use a complete 3D image of the heart to see what the cardiac output is of that heart, and that's a much better biomarker of the performance or the health state of a patient. But now, yesterday, I learned that it's also interesting to see, or was it earlier today, where there's microbubbles present, and actually ultrasound is very good at detecting microbubbles. So maybe we should build a bridge between the ambition of the startup and the ambition of you as a community to monitor what's going on in the heart of a patient, whether it's critical or whether it's in space. And that may be critical as well. Similarly, and Alexander just left the room, one of the things that came to my mind is the COVID pandemic. What did we try to do? We tried to understand who was infected with COVID and who wasn't, and preferably minimally invasive. And one of the elements that I learned by having a quick chat with people that have been active in space is that actually whatever you do in space, you want to deal with it as minimally invasive as possible, because healing processes are quite poor. So this is, again, it's a breathalyzer. There's a microstructure here that captures the particles that are of the relevant size, so that we know that there's likely to be virus in there. And then we can use that as a surface to perform PCR on. And then that's actually double interesting because that surface is also highly thermoconductive so that we can cycle very quickly. The first application here was actually to go to almost space, but for in flights. So whilst you're waiting for your flight, you could do a super rapid PCR test to see whether you have COVID or not. A sample to answer in about 7 minutes. So that's quite good. Again, is it interesting to look at particles that are in the air in a spacecraft to see whether there's an infection or an infection going on or coming up? Probably it is. Then this, I think, is one that actually relates for the people that were in the previous session, where we were interested in seeing how people's body responds over time to being isolated or being in zero gravity. And this is a wearable, so it's not really a point of care, so I'm going slightly off topic. But it's a wearable that fits a microdialysis membrane right under the skin. So there is a level of invasiveness, but it allows us to sample every 20 minutes, a sample from that individual for up to 3 days. And that's the current state of the art. We expect that to be longer, so up to 30 days. And that would allow you to then see what how your biomarkers evolve over time throughout the day, but with very high longitudinal resolution. And that's was already mentioned that's something that we want to become better at. We want to gather much more data so that we can draw sharper conclusions. And actually we have very limited patients. We can't really get more data by increasing our number of subjects that we look at. So then hopefully we can create more data points by looking at more samples over time, which I think is quite relevant. And then one of the other ones, and this is a slide I popped in the last minute, is actually again related to data. This is, of course, on Earth, a big problem, antibiotic susceptibility, providing the right antibiotic to the right infection so that we don't increase antibiotic resistance overall, but also save the life, of course, of our patients. But secondary, it's also interesting to look at the modes that we can measure in different environments. So, so far I see actually a lot of analyticals that are performed in space that are relatively simple, but when you do something that's relatively simple, sometimes also you don't capture all the information that you could capture. This was inspired by a conversation with a microbiologist who I didn't expect on a space event, but then I was naive 48 hours ago. But this is actually a graphene monolayer on which a bacteria or another microbe lives. And whilst it lives, it vibrates, and we can measure the vibrations by looking at this graphene layer. And as soon as we end antibiotics at different concentrations, we can see how quick it stops moving, so it stops vibrating, and therefore we can see whether it's susceptible to the antibiotic. But you can imagine that if you do, my partner is a synthetic biologist, and the experiments they do take quite some time. So if you can say my experiment was successful in 2 hours rather than waiting 48 hours, you'll be able to test much more conditions, and especially as you are very time constrained in the missions that you have, I think this would be relevant to consider. Then this is this is actually an automated allergy test. So it performs about 20 automated pricks, and then it uses AI and imaging to see whether you have an allergy or not. And this is actually on Earth a problem that I think is also in one of your studies a problem, is because simply we are facing a shortage of healthcare personnel and people that can reliably perform relatively complex tasks. And actually this is a relatively simple task because you will get a skin bump, yes or no. I think most of us will be able to say whether we have an allergic reaction or not. Yet this is a successful company for dermatologists because dermatologists simply have a shortage on assistants that will be able to perform the pricks and much rather have a kind of photo booth type system where a patient goes in, gets their analysis done and gets it out and have that in a consistent way. But you can imagine again that if you have a space station where certain routine tasks need to be performed, you don't want that to be performed by a scientist that costs 1,600 euros a day, but simply by a device that can do that for free. Then there's a final call actually I have to you as an ecosystem, and that's to interact more with innovative parties that are active in medtech innovation, biotech innovation. Unfortunately, in these both industries, innovation really happens in small companies. So there is a very, very strong M&A culture. So large biotech companies, large medtech companies will simply pick and select the successful startups and acquire them. That's interesting for VCs, but for us it's a little bit harder because that means that innovation is very distributed. But what I recommend is to try to get in touch with people that are in ongoing development in an early stage, preferably what I call the inventive stage, the feasibility stage, when they start to understand what their product is going to be, but it's not fully mature yet. Because as we develop the demonstrator, it's the kind of the moment where everything comes together and that we can do a first in human or a pilot stage clinical trial. At that point, there's still a lot of flexibility in an organization like that. Also to kind of alter the specifications that they want to work towards. Yes, fine. And also they are very open to collaboration. As soon as they have a finalized pilot clinical trial and they're moving to a prototype that is a kind of, could also consider as a frozen design specifications. The only thing that they worry then is about getting to clinical trial as soon as possible, getting to commercialization as soon as possible, and anything else that comes into their inbox that is not related to clinical trial or commercialization will be ignored. So therefore, I would think that setting up schemes, programs to try to get in touch with early stage startups could be an interesting mutual benefit. I think that's my presentation. I thank you for your attention, and I hope I could have contributed a little bit, even though I have no background in space. Thank you.
I realise we're running late, and I thank you for your patience. I will take one question.
Very nice presentation. Can I ask one question related to the interstitial fluid sampling device in skin? At least that's what I understood your sampling. What is the volumes you can sample, and has it already been coupled by you or others you're collaborating with, if you can comment on that, to anything on the detection side? Because probably volumes will be small, no?
Yeah. The volumes depend on the time that you integrate over. I think would be my answer. So you can get quite large volumes, but then you might lose some of your time resolution. The second answer is it's a sample collection device, so it's doesn't couple to any specific analytics yet. So you can use that sample to analyze in whichever method you are interested in. So that's. Yes, to an extent. I think the application of the device on Earth is a very specific application, so we're not really interested in a broad range of analytics. That said, this is the moment I think that a company is still open to have that conversation and see whether can I continue to mature my technology in function of your application, so that there's a mutual benefit. I can mature my technology and you can use it for whatever you are interested in.
Thank you very much.
Thank you.
I'll leave it. Oh, last but by no means least, I believe we have
The microphone, I keep forgetting. Do we have Ulrich here? We do. Welcome. Ulrich comes to us from DLR, correct?
Partly DLR.
Okay.
Yes.
And he is talking about human blood coagulation in spaceflight-related environments. That's correct.
Thank you and good afternoon. The clicker, the most important. Yeah. Yeah, thank you and good afternoon, ladies and gentlemen. I hope we can discuss a bit if we have overlooked a spaceflight-related risk for the last 50 years or maybe even longer. And this is why I would like to talk to you about blood coagulation. Yeah. What are the basics? So I'm a medical doctor. I'm working as an anesthesiologist. So I'm not aware. I don't know how much you are aware of how blood coagulation works. So just some basics. So it's a natural reaction to injury and inflammation. So we have clear links to immunity and inflammation. So the system helps the body to stop bleeding whenever we injure tissues or vessels. We have a primary part of it. It's done by the platelets, so the thrombocytes, and the secondary part, it contains proteins. It's well orchestrated, and there is a pro-coagulatory and an anti-coagulatory side, and both has to balance very well. And whenever we in clinics we think about blood coagulation, we might Virchow's triad. So it's a very classical concept. And Virchow, he postulated that whenever the blood changes coagulability, the blood flow is disturbed, or the vessel or endothelium is injured, thrombosis or blood coagulation can appear. So my question is spaceflight associated with an increased thromboembolic risk? And we could answer it yes. Why? So we know that just 620 astronauts have been in space so far. And even in this very small group, small number, we know that at least one developed clinically significant thrombosis. And there's another case, second one, that at least in a retrospective analysis of ultrasound images, it seemed that there was a thrombosis as well. So we know thrombosis appears in space. We know as well that it appears very late in space. It happened at around 50 days during a long-term mission on ISS. So it's too long that we can say it had something to do with factors that took place before the start. And it was a very unusual location. It appeared in a neck vein. So usually humans on ground on Earth, they develop a thrombosis in the veins of the leg or of the pelvis. And when we remember the triad of Virchow, this shows you this image, we find, at least in a theoretical way, we find all these factors back in space. So we have endothelial dysfunction caused by the stressors we have discussed during the last 2 days, hypoxia, radiation, psychological stress, decompression, all these things. We have blood stasis or even reverse flow, especially in the veins and the vessels of the neck and the head. And we have hypercoagulability because the astronauts, they lose fluids, they concentrate blood, and they concentrate the coagulation factors as well. So from a theoretical standpoint, we can say, yes, everything is there to develop thrombosis in space. Furthermore, a space mission does not is not only weightlessness and being in space, but also going up and landing. And these phases are characterized by high gravity loads. So we got the opportunity already more than 10 years ago by ESA to perform blood coagulation measurements on a long-arm human centrifuge in healthy individuals, 20 male subjects. And we measured how their blood coagulation capacity changed during a 15-minute run of 3G in sitting position. Oops, sorry. Here. And what we saw is on the left you see the clotting time that took native blood, so let's say normal blood, to start building a clot, building a clot, and it took the clotting time was significantly shorter at the end, especially after 30 minutes of centrifugation compared to start of centrifugation. And as an indirect marker of coagulation, the D-dimers, so there are breakdown particles of blood coagulation, they were increased. So we can say hypergravity at least is associated with increased blood coagulation capacity. I ask the question again, is spaceflight associated with increased thromboembolic risk? And I say no. Why? So we know from patients with spinal cord injury that are chronically immobilized, that they regain after a while after trauma thrombotic risk that is equal again to general population. And you know, astronauts, for me as a medical doctor, is an immobilized individual as well. So I see parallels and similarities in these two groups. I know that thromboembolic incidents have not been observed during bed rest studies. So one of our best modules for pathophysiological changes in space on ground. People are immobilized for 20, 30, 60 days. And again, 620 individuals lived in space in a theoretically highly thrombogenic environment, but we do not have clinical accounts on thromboembolic events in astronauts. Let's say, so the typical signs of an embolism is you have a swollen leg, you have a swollen arm, it changes color and it hurts. So as far, no of these incidences have been published. And on Earth, also 1 to 2 individuals from 1,000 per year develop thrombosis. So it's not that you don't do it on Earth. And we remember the location was very unusual in astronauts. Even this is possible on Earth, even in healthy and fit individuals, and we call it Paget-Schroeder syndrome. So it's affecting healthy individuals that are heavily exercising, especially with the upper trunk, weightlifters, baseball players, and so on. And when we imagine how an astronaut lives in space, I think he or she will do much more with the upper body than with the lower body compared to Earth. There was a study published last year that found a mechanism in mammals that is highly conserved about across different species, that nature has natural thromboprotection. So the body does not do nothing when it is immobilized, but there are mechanisms that can be switched on. These mechanisms were first found in bears, because these scientists, I don't know how they got the idea, but they asked themselves, why do not die bears during hibernation during the winter of lung embolism and thrombosis? And then they found a very interesting molecule they call the heat shock protein 47, and it's the regulation changes much during winter and during summer in bears. Interestingly, exactly the same protein changes in patients with spinal cord injury as well. And this is why we as space doctors come into this game. We try to find the same mechanism in a bed rest study in 2022, and exactly the same happens in our subjects in the bed rest study. These individuals also decrease the expression of heat shock protein 47 after 27 days of strict bed rest. So the question is, does an astronaut have this mechanism as well? And this is why I'm very happy that ESA supports our experiment that we got accepted already a couple of years ago, and we call it space hemostasis. And we would like to really look into these mechanisms of blood coagulation in space. And we will cover not only weightlessness, not only the phase in space, but also the phases prior the missions and especially after the mission, because we know, for example, from economy syndrome that thrombosis and embolism sometimes developed even with a latency, so after a while, after the causing stress. So what we will do, so we will measure astronauts during long-term missions, and of course we will try to perform vascular ultrasound on ground. Maybe in space we will see medical questionnaires, point of care coagulation testing, subsequent coagulation tests in our laboratories, and proteomics. So that it works, these are data from the currently running bed rest study at DLR, so 60 days. On the right, you see data from function testing of platelets, of thrombocytes, and we try to find whether the function or the clotting capacity of platelets decrease after a while in bed rest. You see here -5 BDC after 3 days and head until 10 days, 20 and 60. So 60 days is just over today. And indeed, maybe there is a tendency that platelet function decreases after a while in bed rest study. So we will look into this in much more detail. Also, we go to patients, back to patients, and we will investigate whether radiation makes a difference in blood coagulation. And there is a very interesting group receiving radiation exactly in the area where we found the thrombosis in astronauts at the neck. And these patients have cancer at the neck, and they receive proton therapy exactly there. There is a very specialized clinic in Essen in Germany. And in general, you will start to investigate these patients before and after the therapy with proton beams, and to look if blood coagulation changes. So beside normal laboratory testing that is done subsequently, we have indeed point of care testing. These are well-established methods at the hospital in the clinical work. But you see all these machines, they work with normal laboratory equipment. You have to handle the blood, you have to put it in little reaction tubes and so on. All these machines are not space ready at the moment. And the question is, do we need such machines modified for space to investigate blood coagulation further? Personally, I must say I'm not convinced yet, but maybe we can discuss this. Okay, so what are our next steps? So we would like at first to phenotype blood coagulation in space on ISS and over the duration of a long duration mission. Of course, we would like to execute similar studies in relevant space medicine backgrounds. I think the topic of natural thromboprotection, this is very interesting, and I must say at the clinic nobody talks about it, so nobody knows about it, and it would be really interesting to see if there are any countermeasures we can develop in this space community to maybe foster these natural protective mechanisms to decrease thrombotic risk. So we will participate in further background studies, and today or yesterday there was a discussion about the length we need for background studies, and the question was, do we really need 60 days? And I must say yes. For blood coagulation, to understand these effects, we need long studies, at least 30 days, better even longer. And yes, the countermeasures I have already mentioned. And this is my last slide. And I thank you for your attention. So what do you think? Risk or no risk?
I think it would be great to measure HSP47 in the blood using an immobilized antibody on a microfluidic system.
Yeah, so we need the machine from Munich.
And I also think that machine should be miniaturized, so that we can fly it and fly it ultimately to the moon and one day to Mars. Ladies and gentlemen, I realise we're running late. Thank you for sticking around. Does anyone have what's the thing? Oh yeah, questions. Does anyone have a question?
Yeah, of course.
Thank you. Fascinating talk. And one of the endogenous protective factors may be nitric oxide, but that's well known anti-thrombogenic, and it's known to be upregulated in hypoxia in response to all sorts of stresses, right? So maybe that's what keeps us going. And with microgravity, there is it's easier to keep the heart's function pushing the blood around, but there is less help from the lymphatic system due to lack of muscle action, right? So I was wondering how that topic might change with in the context of devices. I got ahead of myself. In the context of exercise as a countermeasure, which is taking 2 to 2.5 hours per day, I understand in some cases for astronauts. So does that help? Is that an additional stimulus for antithrombogenic protection? And or can it get much worse when on longer missions the capability of mounting that response drizzles out? So if astronauts become increasingly tired, the duration of a mission extends, and they're actually no longer capable to mount that physical activity as they usually would do. what happens then? Nobody knows, but I'm asking, is there anything in the literature you have looked at in a more chronic setting that would predispose patients over a longer period of time, in particular those with endothelial dysfunction? So if you think of obesity, diabetes, chronic ailments we are familiar with, that predisposes to an increased thrombotic risk.
The answer is yes, of course. We know age, for example, is a very important risk factor. So the risk increases just by getting older. Diabetes, of course, and overweight, all these classical risk factors. Exercise is an interesting topic. So acute exercise increases at least the capacity of blood to coagulate. So at DLR, what the astronauts don't get is they don't get a screening for genetical risk of developing a thrombosis, but our bed rest studies, they do. They get it. So we do a genetic screening, and we have done this in the past just after doing an exercise test. And then we send all this blood to our laboratory, and we ask, does this subject has an increased risk? And we very often got the answer yes. And we were very surprised why. And what we learned later is that several blood coagulation factors, for example, factor 8, is increased after intense exercise. So, of course, exercise on a short term increases your risk, but on a long term, as you said, yes, your vessels get smoother, you maybe produce more NO. It should be protective. Yeah. There's a general problem that we do not have enough long-term studies, and studying these effects in a very controlled and detailed investigated population. Yeah.
Are there any further questions from the audience?
In which case, I will wrap this up. Ladies and gentlemen, you have heard about coagulation. You have heard about immobilized antibodies and doing chemiluminescence tests. And you have heard about the miniaturization of point-of-care devices. This brings to a conclusion day 2 of the HRE workshop. Have a wonderful evening. Enjoy a Polish beer or 3, or indeed some Polish vodka if you haven't already found it. And I look forward to seeing you in a plethora of sessions tomorrow. Thank you very much, and thank you to all of those online who are still here. Thank you.